Good day, my dear postgraduate students. Welcome to the session on Glide the Slide. In the last class, I had given you a homework on a case history with a 10-year-old boy developing diplopia, and this was the histological picture. As I had been telling you people, before ever plunging into the diagnosis whatsoever, observe. In fact, the authors, I was bewildered at their depth of knowledge. They are not telling me anything about something that is very striking, but instead they are concentrating on some cells which are having a pinkish, somewhat an epithelium-like picture. So these are the epithelioid cells. And then there is some kind of a ground substance that is supporting the cells. And last, we have got this large cells with bubbly cytoplasm. Glide the slide 14. I hope you people are able to come to a diagnosis. I'm able to see clear cells. I'm able to see epithelioid cells. I'm able to see cells with bubbly cytoplasm. And there is, so the diagnosis is chordoma. It is a low-grade, locally invasive, and locally recurrent tumor. It arises from the remnants of the notochord. That is why it is called chordoma. And it can occur anywhere along the spinal cord, but the most common site is 50% can be at the sacral region, 35% at the base of the brain, and another 15% can be in the mobile spine. The 5-year survival rate is about 50%. The etiology, they say, is the mesodermal structure in the embryo that gives rise to the notochord. Later on, it becomes redundant and the nucleus pulposes, which can cause a prolapse of the intervertebral disc. The genes implicated are the brachyury gene, mechanistic target of rapamycin gene, mTOR, tensin homolog, P10 deficiency. Usually it occurs in the age of 40 to 60 years of age, and the male-female is 2:1. 20% of the spinal cord tumors are constituted by this, and 3% of malignant bone tumors. The classical clinical history will be like this. It depends on the location, wherein there can be a deafness, a headache, a cranial nerve dysfunction, and it can also occur in the sacral region, causing a localized pain or a bladder dysfunction. This can be the clinical history. Grossly, you find it can appear gelatinous or hemorrhagic. The modes of diagnosis can be X-ray, CT, and the MRI. There are 4 variants in it. In fact, 3 are there, and the 4th one I had mentioned as chondroid is supposed to be a subvariant of the conventional. So I can mention it as 3 with 1 subvariant. Conventional, poorly differentiated, dedifferentiated. And remember this, chordoma, brachyury gene will be positive. I would like you people to again describe. So the bluish matrix again, it can be chondroid and the pinkish region can be myxoid. I'm finding this vacuolated cells almost resembling the chondrocytes. The importance of it I shall be telling you. And there are cells with large vacuolation and they are in fact bubbling with exuberance. Thanks to Science Direct for this wonderful photomicrograph. And web pathology similarly for this. The tumor has got a bosselated appearance. It can infiltrate into the adjacent tissue. Recurrence is quite common, local recurrence. At the same time, it can be solid, it can be cystic. And I'm able to see a gelatinous pinkish hue or a bluish hue and shiny appearance, which again is one of the characteristics of a chondroid tumor. So that has to be borne as a differential diagnosis in mind. The differential diagnoses are meningioma and plasmacytoma because of the location, clinically, nothing to do histologically. But then chondrosarcoma, pituitary macroadenoma, metastatic carcinoma, myxopapillary ependymoma, echodosis, which is a hamartoma, can all be the differential histological diagnosis. The markers for this are cytokeratin cocktail, CK8, 18, and 19. Epithelial membrane antigen and then S-centered protein are positive in conventional type. P10 loss can be seen in this as a negative mark for diagnosis. Thanks to web pathology mentioned over here, I find that there is a matrix that is showing a degeneration, myxoid or a chondroid one, and then there are the bubbly cells over here. And this is the sacral region showing a large tube. Classically, how do you describe the classical type of it? It is lobular with separating fibrous strands. Within the lobules, there can be cords, nests, and epithelioid sheets. Cells can also be single. The epithelioid and the clear. Epithelioid is something like the epithelium. Clear is because of glycogen. And because of this, there can be eosinophilic to vacuolated cytoplasm. Large, bubbly, physaeliferous cells. Extracellular myxoid matrix. Nuclear pleomorphism is there in the high-grade areas. The nucleus is otherwise vesicular. Similarly, areas of necrosis and mitosis can be seen in high-grade tumors. These are the variants. Chondroid chordoma will be having a picture of a cartilage. Usually occurs at the base of the skull. Dedifferentiated means it is biphasic. Usually I will be finding a very clear cell resembling the chordoma, whereas there can be the spindle cells also. This dedifferentiated can occur in the liposarcoma also. Well differentiated with an abrupt area of variation. Poorly differentiated, I'll be finding epithelioid rhabdoid cells, mitosis markedly increased, necrosis seen, no physaeliferous cells, and not much of stroma. So these are the things I have already mentioned. Look at this one. I find that cells are well differentiated and they are having the vacuolated cytoplasm. All of a sudden, I am finding that the cells are becoming poorly differentiated. And look at the mitotic figures over here. There are so many pictures of the mitosis that is there. And there is hardly any of the matrix that has been mentioned in the other types. So the conventional going in for a poorly differentiated. Let us glide to the next slide. A 5-year-old boy with diminution of vision and an awkward gait. What is your diagnosis? Always describe. Diagnosis is secondary. Thank you. Meet you in the next class.